Okay, welcome to Mission Bio's office hours. Today we are going to be focusing and talking about the Tapestri instrument, as well as some general guidance around cartridge handling, as well as emulsion handling when using our product. The two main documentations we have available on our website for the instrument include the Tapestri Instrument User Guide, as well as the Site Requirements Guide. The Site Requirements Guide typically is helpful for you to understand what are the minimum requirements for you to set up the instrument when you first receive it. You'll notice it doesn't have any strict requirements. You can place it anywhere in your lab on a stable surface that is not vibrating. And we recommend it to be not too close to a venting system, as any microfluidics technology may suffer from a potential debris that's in the air and might enter the microfluidics device and cause clogging. So just be aware that you don't place it on a table that has a centrifuge, and otherwise you can move it along as well without having to reinstall it every time. The user guide itself talks a little bit about the firmware, as well as some maintenance steps that you can follow in order to keep the instrument in good shape. The instrument as well as the cartridge are listed here in this on this slide. From an engineering perspective, it's a very simple construct, which comes with benefits because there's not that much that can go that can break. So we are very pleased with the overall rate of instruments that we have in the field that are supremely stable. You see on the left-hand side, it's about the size of a larger PCR machine that contains a lid that you would close and open in order to start programs. You have a touchscreen, 7-inch, your typical iPhone touchscreen in terms of its size. A USB port on the front panel, or in some instruments, it's also on the back panel, which you'll typically only use if we have a new firmware ready to roll out for you to update, or if you need to export log files. So those are just text files that include diagnostic data for us to troubleshoot if necessary. On the right-hand side, you see the key and sort of the heart of the entire technology, which is the cartridge. The DNA cartridge, to be accurate, which is shown in transparent. It sits on top of the base plate. So the cartridge is the microfluidics device itself that contains a lot of reservoirs for reagents as well as the microfluidics channels. The base plate on which it is mounted to contains a number of slots that are necessary for you to install tubes, collection tubes, in order to collect the emulsions that are being generated. And on top, you see a gasket, or the DNA, the cartridge gasket, that will facilitate the pressures to be able to be generated so that the instrument can start working. Some of the key elements in terms of some of the key differences across our, I think right now, two versions of instruments that we have are just listed in this slide. So some of you might work with our first generation. It's been in the field for a very long time. It does not contain the integrated UV lamp. That's different to our new generation instrument, which comes with some cosmetic changes, as you can see here, in terms of its color, but it also has an integrated UV lamp. What that means is that as you are barcoding your sample, instead of running it just for 35 minutes, it actually runs for 45 minutes, as it now consolidates both the actual barcoding program plus an additional 10-minute step at which your sample is being exposed to UV light. Just simplifies the entire workflow, and you don't have to handle your emulsions outside of the instrument unnecessarily. The USB port, again, as I mentioned earlier, is a slight change in terms of its location on the front panel here and on the back panel for the newer instruments. Firmware, again, very simple. It contains two main programs, encapsulation and barcoding. The barcoding program actually is partitioned into two separate programs that we refer to as priming as well as barcoding. And then also be aware that every program that you'll start, the instrument will sort of run a short initialization test for about 5 seconds, where it tests whether or not pressures can be built up. So it's looking for a leak, and if there's a leak for whatever reason, maybe you misaligned the cartridge, the gasket, it's going to give you an error. So that's just important for you to know that as you operate the instrument, that you don't go away immediately after you start the program. You want to spend 5 seconds or so to verify that it actually goes past the leak check test. For those of you who have worked with the instrument, this is just a sequence of screens that you'll see on the touchscreen as you start the program. And this is an example for encapsulation. So you start your program, it's going to ask you whether or not you really want to start. This is where it initializes for about 5 seconds, and then it starts the program encapsulation. When we talk about barcoding and how it's partitioned into 2 steps, and that's illustrated here in this slide. So the barcoding program includes a initial first priming step that really enables you to fill parts of the microfluidics device, in this case, microfluidics electrodes with electrode solution. Previously, this is a screenshot of the cartridge. So we're looking with a microscope inside the microfluidics channels architecture. Those are initially filled with air. You fill them with an electrode solution so that you have a carrier in order to carry voltage. And the reason why we do this is now because we are having, we are having a number of reagents being part of the barcoding program, one of them being your single cell lysates. So those are encapsulated single cell lysates in drops. Drop with oil with water typically doesn't mix well. And so in order to be able to combine our contents from our single cell lysates, which is liberated single cell DNA, with all the other reagents necessary for barcoding, we, in a very controlled fashion, destabilize the surface tension using a voltage that we can now do with the electrodes. So that's why we sort of prime the cartridge, make it ready, get it ready for the actual barcoding program, which is then shown on the right-hand side. That includes the reagents that you're probably used to, including your barcoding beads, your single cell lysates, as well as polymerase, DNTPs, and other reagents. This just shows, I think, yeah, this just illustrates the initialization step. So you start a program, encapsulation, for an example, it runs through about 5 seconds. It's checking the seal, and if it doesn't detect a leakage, it's going to move forward with the actual program. Okay, there's minimum maintenance for the instrument, which is also a huge plus. The only maintenance that we recommend is for you after barcoding is done to clean the electrode pins. And it's, I don't know if it's shown, I have an image later, but the electrode pins are on the bottom of your lid, and they are in direct contact with the electrode solution. So that's the only part of the instrument that's actually in direct contact with any of your reagents. And so because it's a salt solution, we want you to just clean it up, ideally after every barcoding, using dust-free wipes to prevent potential buildup of salts. So if you don't do this, they will look white at some point, and it might render the electrodes useless. So just a quick step, 10 seconds or so to clean them. Okay, moving on to cartridge handling, which is important, and some of those bullet points are not news to you if you are used to microfluidics, a microfluidics environment, but it's paramount that once you handle the cartridges, that you prevent them from the environment as much as possible. So when you unpack them, and really only expose them to the air if necessary, which really means if you actually load reagents, otherwise just have them covered. And we typically recommend to always operate inside the instrument. So as you load your instrument, as you load your reagents, don't do this outside of the instrument, but rather inside the instrument. And if you don't use it, just simply lower the lid. Base plate mounting, that's something you do the first time when you actually install the instrument. So it's a pretty straightforward process. You just have to make sure that number one, that you hold the cartridge by the sidewalls, so never touch it on the inside of the reservoirs. And then also when you work with samples, that you make sure to only use our validated emulsion safe collection tubes, which come from a specific brand, in this case Oxygen. They are optimized in order to not just minimize what we call droplet merging, so the combining of drops that you want to prevent in order to preserve the single cell aspect, but also they can sustain the high temperatures that are that you will experience during thermocycling up to 95 degrees. So it's important to not replace those with your generic PCR tubes. It's straightforward to mount the cartridge as well as the gasket. As you can see, there are different ways to do this, and only one way is correct. So this is just the top view of the cartridge. As you can see, the reservoirs, those are the reagent reservoirs, they're asymmetric. So the majority of our customers just use the reservoirs in order to orient themselves. Some customers also use, on the left-hand side, you see sort of those posts. They're different size. So we have a long post on the top left that has to be pointed to the top left corner. So if you try to mount it that way, that's not gonna work. In terms of the gasket that you apply on top of it, you now have 4 different ways to do this. And again, similarly, it's hard to do this incorrectly because you have, again, the notch, sort of this notch, which is sort of this punching that will sit right on top of this long post, as well as you have those little holes here that you notice that they will need to go through the electrode pins on the left-hand side. If you, for some reason, do it that way, you will notice the instrument lid does not close. And then sometimes you'll see that you know, the gasket may not sit on top of the cartridge perfectly aligned. So that will likely cause an error on the instrument side when it's looking for pressures, if it can hold pressures. So here on the right-hand side, you see that it's slightly misaligned. On the left-hand side, it should fit perfectly on top of the cartridge.
Hey, Robert.
Yes.
We have a few questions from the audience, so I thought I'd interject since it was pertinent to this point in the process.
Yes.
Great. So if the initialization check fails, what can I do to solve the pressure error?
That's a good question. So if that test throws an error, it could have multiple root causes. One of them could be, just as I pointed out a second ago, a misaligned gasket. So it's worth opening the lid and then verifying that the gasket is perfectly aligned on top of the cartridge. A second potential root cause could be, in very rare cases, a delaminated cartridge, so that it starts, if you will, the PDMS sort of starts breaking up, and then there is a small leak. Whatever pressure is applied will escape that. We've seen this only very, very sporadically, but there are ways, when you get in touch with the support team, there are ways to really pinpoint and narrow down the root cause by using another used cartridge, for instance, to see whether or not it can hold the pressure. And if it can, it likely pinpoints to a cartridge failure. I think those are the two main causes. Sometimes there's debris on the gasket. So every kit that you purchase comes with its own designated gasket for each sample. We would like you to keep those dust-free, as they also could contribute to potential pressure issues.
Fantastic, thank you. And then one more question we have here. Can I image my droplets and beads on the tapestry as you showed in, I think, a slide coming later on?
Oh, I see. You mean this one?
Correct.
The answer is no. We obviously here at Mission Bio at R&D, we want to take a look at that. We want to investigate that process as we are continuously optimizing that process. There's no benefit for you as a customer to visualize that, as there's nothing you can do about it. We also don't want you to take emulsions that you collected in an emulsion tube and visualize them under a microscope, for instance, as this would require you to go into the emulsions and manipulate them. Every time you go into the emulsions with your pipette tip, you will cause some small mergers. So you compromise the quality of it. Yes, and this is also an FYI, this is an old architecture. So, you know, the cartridges that we have today are looking very much different in terms of its channel architecture. This is really just to illustrate the concept of what we mean by priming versus barcoding.
All right, thank you very much.
Mm-hmm. All right, moving on. Again, this is just a nice reminder, really good practice to, when you load your reagents onto the cartridge, either during encapsulation or before priming or before barcoding, we highly recommend to do this inside the instrument. So don't have your base plate and the cartridge sitting outside, because number one, it's again exposed to the air. So even though you don't see anything in the air, there might be microscopic dust or debris that might just fall in. And then secondly, some of the reagents that you load fill up really all the way to the top. And so it becomes challenging to move that from one place to another without spilling your reagents. And that might compromise your experiment. In terms of pipetting, nothing new here, only that we always recommend you want to minimize bubbles, which you can do by just going all the way to the first stop of your pipette, not to your second stop. Typically two stops on your pipette. So only go to the first step, and then secondly, you want to leave space between your pipette tip and the inside wall of your reservoir. That's not so critical for PCR mix and the generic reagents, but it's definitely critical for the emulsions when you pipette them, as well as your cell suspension. Otherwise, particularly for the emulsions, if you set your pipette tip to the very bottom, the emulsions throughout this process of pipetting will potentially break. And then you are unaware of that, and your single cell aspect is unfortunately ruined, and you will compromise your experiment. If you do see bubbles, which sometimes it's unavoidable, some of the reagents are just very prone to produce bubbles, simply poke them away with a clean pipette tip if they're on the surface. If they're at the bottom, you definitely want to get them, you want to remove them with a clean pipette tip. Some other recommendations, again, if you load reagents, some of the reagents are very, very prone to exit your pipette tip. One of them is the oil. So encapsulation oil or barcoding oil tend to flow out of your pipette tip if you hold it vertically. So in order to minimize potential contamination, we just recommend to go around as you pipette those individual reagents, as opposed to just going across. And then again, when you apply the gasket, don't push on top of it. There's no need to push with force on the gasket when you try to apply it onto the cartridge, but just lay it on top. If you push, there's a risk that you potentially push too hard so that some of the liquid that's already pre-filled in the cartridge will touch the underside of your gasket, and then this would be not ideal. Something just to keep an eye on: if you open your lid, you may have noticed sometimes the gasket just sticks on top. It's not a big deal. It's just sometimes captured by the electrode pins. So just remove it when you're done. That's a good practice to always remove it if you open the lid, once you open the lid, so that you don't forget about it when you close it again the next time, or that you don't accidentally put another gasket on top and you wonder why the lid doesn't close. More about emulsion handling. That's very critical. As soon as you create emulsions, you want to preserve their single emulsion format. You want to minimize mergers. So the process of multiple drops fusing and merging together, which typically happens either mechanically. If you drop a tube, then most of your drops are probably merged, or if you are electrostatically charged. So we want you to not to be in direct contact with the interface of the plastic that is associated to your emulsions. So hold your tube by the lid. And then also when you close your lid, I know it's extremely routine when you close a PCR tube or an Epinephrine tube just to do it that way by holding the base and then just closing it. We don't recommend that. Always have a PCR, a 96 well PCR rack handy, so you can place it on top and then just use one hand to close the lid. Really just precautions to improve your data quality overall. The emulsion quality, again, everything that looks white, those are your millions of drops that are being generated. The image on the left is what you are supposed to see. The image on the right, in very rare occasions after encapsulation, you see something like that. In that case, just simply contact support and we can help you address the issue. But that's what we typically refer to as jetting, which might be the cause of debris in your system or at this site where the drops are being formed. Where if there is debris, then it will likely not be successful to generate drops. And then I know we are running short on time. Emulsion handling, as you will review the user guide, there are 2 steps that require you to remove oil prior to thermocycling. And as you see on the right-hand side, we are using gel loading tips to do so. Instead of the regular pipette tips, we pipette very slowly in order to not have too many emulsions dry out on the sidewall that are being left behind. And then ideally only go in once and then go out once. Don't try to go in multiple times to remove oil. And just as a reminder, the whole reason why we do this is to lower your overall volume to 100 microliters so that it is adequate for your heat block on a thermocycler. And the emulsions are really the part that we are trying to temperature control. Barcoding, a friendly reminder, this is how your barcoding result looks like. You will see some variability across all 8 tubes. This is the step where we partition our sample into 8 replicates due to volume restrictions. You will see some variation, which is perfectly normal. You sometimes will also see a tube maybe that's not filled. That happens in very rare occasions, but it typically means that maybe that was a microfluidics channel that was blocked, and then the volume would be separated across 7 tubes instead of 8. So it does not typically mean you lost signal or you lost data. And then similar.
We have a question from an attendee. If some of the oil, some of the emulsion was inadvertently removed during the oil remover, can they pipette it back in the tube? Would you recommend that?
That's a good question. We do not recommend that for two reasons. Number one, as you pipette emulsions in and out, you likely will increase the fraction of merged drops. And those are drops, again, now that may contain multiple beads, barcoding beads, and or multiple cells. What that means is that you will definitely generate a product you will generate library, but it's a sort of a semi-bulk context. And so that will impair your Tapestri pipeline on the bioinformatics side to clearly distinguish those from true cells. So if you do suspect that you made an error, or if some of the emulsions look extremely low on quality, meaning that they have this transparent appearance, then we recommend removing that tube from your downstream analysis to not combine with the rest.
Thank you.
Okay. This is just, again, a screenshot. As you can see, just to the question from the attendee, we don't ask you to remove all the oil so that you actually risk potentially removing emulsions. All we ask you to do is go with your pipette tip all the way to the bottom and remove as much so that you have less than 100 microliter. So it's perfectly fine to have 30, 40 microliter of oil remaining in the tube. And then lastly, and this sort of speaks to just your thermocycling step. After you thermocycle your emulsions, you want to make sure and you want to ensure that your emulsions look identical in terms of its phenotype and appearance compared to before you thermocycled. So you put your emulsions on the thermocycler, you typically run the program overnight and you come back the next morning to assess its quality. If you see something like on the left-hand side, that is concerning because that indicates low quality of emulsions. All the emulsions essentially broke in that part, which means all the content was released and it looks aqueous. In this case, all the emulsions are intact. And so again, this typically never happens unless there is some issue with the heat transfer on the thermocycler. So we have recommendations in the protocol that you're supposed to use a skirt or corner tubes to address that. Or sometimes if a tube wasn't closed, then you would also encounter something like that. But again, if you see something like that, if any of your tubes has an appearance like that, please discard that tube and do not continue with that downstream. All right, that concludes that little presentation around Tapestri Instrument and cartridge and emulsion handling.
Fantastic. Thank you so much, Robert. I think we have just one more question that we didn't get to during your talk. The question is, can I store my Tapestri in a biological safety cabinet if I'm working with biohazardous source material?
Can I store, sorry, the first subject, can I store what?
The Tapestri instrument in a biological safety cabinet.
Yes, I believe that is possible. In the site requirements guide, you will see some requirements in terms of temperature and altitude and humidity. Those are not any different from a PCR requirement, PCR machine requirements. So yeah, the answer is yes.
Great. Well, again, thank you to all of our attendees that attended this seminar. We're happy to answer any further questions. If you have any, please email us at support@missionbio.com. And thank you again to Robert for this really enlightening seminar.
Great. Thank you, Kelly. And stay healthy and safe, everybody.